Dear students, we have started a discussion on Unit 1, that is the metabolism. So in that we have discussed up till now. In case of metabolism, we have studied types of metabolisms, phase 1 metabolism and phase 2 metabolism. So we have classified detail about phase 1 metabolism, what type of reactions coming under this category that we have discussed in detail. Now, in last lecture only, we have discussed about phase 2 metabolism and their conversions. Phase 2 metabolism is mainly associated with synthetic and conjugation type of reactions. So phase 2 metabolism involves glucuronide conjugation, then amine conjugation, and so on. So this type of reactions we have already discussed in the case of phase 2 metabolism. Now, today we are going to discuss factors affecting drug metabolism and their stereochemical aspects. Now, before we move towards the factors which are affecting the drug metabolic studies, we will classify those factors into three main categories. Now, what are those factors which will affect drug metabolism? Now, what is drug metabolism? What are the phases of drug metabolism? Phase 1 metabolism, phase 2 metabolism. This we have discussed in detail. Now we are discussing what are the factors associated, or they are going to affect the rate of drug metabolism in our body. And those factors are divided into three main categories. Those are chemical factor, biological factor, and physicochemical properties of the drug. Now, this physicochemical properties of the drug we have discussed in detail in case of unit number 1.2. So here we have discussed what are those factors: ionization, solubility, partition coefficient, then hydrogen bonding, chelation, bioassociative parameters, optical and geometrical isomerism of the drug molecule. So all these physicochemical properties, they are no doubt affecting the drug metabolism. This we have discussed already. Now we are going to discuss these two main important factors, and those are chemical factors and biological factors. Now, what are those? The chemical factors are divided into two types: enzyme induction and enzyme inhibition. Now, enzyme induction means the enzymes which are going to induce the metabolism of the drug, or which are actually involved in the metabolism of the drug. Now, how the enzyme induction is going to be take place, or what are the enzymes which are going to induce the metabolism of the drug? So generally, metabolism of the drug is the first order process. So the first order process, which means that the constant fraction of the drug molecule is metabolized in the unit time. At a particular time, main or the something amount of the drug is metabolized at unit time. At a particular time, this much amount of drug is metabolized. Now, the saturation of the one metabolite pathway may allow or may change the metabolic pattern of that drug molecule. So they are going to affect the drug metabolism. Now, what will be the effect of enzyme inductions on the drug metabolism? Now, we all know liver is the main site for the metabolism of the drug molecule, and including liver, there are other sites which will involve in the metabolism of the drug molecule. Those are placenta, skin, lung. So they are going to also involve in the metabolism of the drug, but majority of the case, liver is the organ which is involved in the which is going to play the important role in the metabolism of the drug molecule. So the hepatic microsomal enzymes which will do these enzymatic or drug metabolism. Now, the drug metabolism from the enzyme induction may results into either increased rate of synthesis of new enzyme protein. See, when the enzyme induction will be done, so it will either increase the rate of synthesis of new enzyme. See, the chemical factors are divided into enzyme induction and enzyme inhibition. Biological factors are divided into age, the age of that person, diet, what type of diet that person is going to take, sex differences, whether the person or the patient is male or female, then species differences, in which area they are going to reside, then the strain differences and altered physicochemical or physiological factors. So these many biological factors also will affect the drug metabolism. Now we have already discussed that a chemical factor is divided into enzyme induction. Now what is the enzyme induction process? It is a phenomenon of either increased drug metabolizing ability of the enzyme by several drugs and chemicals, as enzyme induction is the agent which bring about such an effect are called as enzyme inducers. Enzyme inducers म्हणजे अशा पद्धतीचे drugs or the some chemical molecules are there which will induces or which will increases the enzyme induction process. Now, what are these mechanisms to induce the enzyme production? Those are increasing in the liver size and liver blood flow. See, we all know liver is the main organ which is involved in the metabolism of the drug molecule. And now why liver is involved in the metabolism of the drug molecule? Because liver will contain the majority of the enzymes which will metabolize the drug molecule. Now, if the increasing in the liver size or liver blood flow, it will increases enzyme induction. Increasing in both total and microsomal protein content, they are going to also affect the increases the enzyme induction. Then increased stability of those enzymes. See, we all know enzymes are very specific in nature and they are temperature resistant or temperature they are unstable towards the temperature. Then they are going to also affect the processes by the presence of another chemicals. So if we are going to increase the stability of the enzyme, they are going to also affect the enzyme induction, increase stability of the cytochrome P450 enzyme. Now, the cytochrome P450 already we have discussed in slide number 1 of unit 1. This is the main enzyme involved in the metabolism of several drug molecules. So if we are going to increase the stability of this enzyme, ultimately enzyme induction process will be done. Decreased degradation, that means we are increasing the stability and proliferation of smooth endoplasmic reticulum. Now there are consequences of enzyme inductions. So what are the consequences which will increase the enzyme induction process? Decreasing in the pharmacological activity of the drug, because enzyme will easily metabolize the drug molecule or immediately it will metabolize the drug molecule. Increased activity where the metabolites are active. So examples of enzyme induction. What happens when the enzymes induction will be done? Now, if a person is going to take oral contraceptive, that is steroids, and this enzyme is going to metabolize that cytochrome oral contraceptive. So what happens? The cytochrome 3A4 will metabolize this contraceptive into inactive and it will be get excreted out. But the agent or the drug molecule which will increases or which will induces the enzyme induction process of this enzyme. Which enzyme? Cytochrome 3A4 enzyme. So this rifampicin will induces cytochrome 3A4. So what happened? See, it will induces and convert them into an inactive compound and excreted out. Now second example is acetaminophen. What happened? The cytochrome 2AE1 will convert the acetaminophen into paraquinone imine, which is toxic one. Will be done by the process of ethanol will induces this enzyme. So these are the effects of enzyme induction. What happened? This enzyme induction will increase the metabolism of the drug molecule and convert them into respective metabolites. But the stimulation of the drug metabolism will result into three processes. Either it will increase the rate of synthesis of new enzyme, or decrease rate of degradation of the drug metabolizing enzyme, and increasing in the amount of smooth endoplasmic reticulum, which is going to serve the site for drug metabolism. So they are there. Now these are the examples of the enzyme induction processes. Now the examples of enzyme inhibition processes. A decreasing in the drug metabolizing ability of the enzyme is called as enzyme inhibition. Then now we know the enzymes are going to metabolize the drug molecule, but the drugs which will inhibit the enzyme process of metabolism, this process is known as enzyme inhibition. And the process of inhibition may direct or indirect. The process of inhibition may be direct inhibition they are going to do, or maybe indirect inhibition. See, enzyme inhibition will decreasing in the drug metabolizing activity of the enzyme. We all know enzymes are going to metabolize the drug molecule, but the substances which will inhibit this process of metabolism of enzyme, those are known as enzyme inhibitors. Now they are going to act by two different ways, either direct inhibition or indirect inhibition. Now direct inhibition will involve competitive inhibition or non-competitive inhibition. Now we all know enzymes are very specific in nature. So, and they are going to act, or they are going to show the processes by their lock and key model. They are going to, substrate is going to fit into the cavity structure or the structure of the enzyme molecule and going to show the effect. Now what happened? The enzyme which is going to inhibit the process of metabolism, which we can call them as a enzyme inhibitors, they may be affect, or they may be showing their activity by the direct inhibition. Now how they are going to affect the direct inhibition process? By competitive inhibition. Now see, this type of structure or this type of cavity, it's provided by this enzyme to attach the substrate molecule. Now this is the substrate will going to show the pharmacological action, but instead of substrate molecule, some another chemical or some another inhibitor will come in and going to attach there in the cavity. So he will not going to show the activity, that is pharmacological action. This inhibitor molecule will compete with the substrate molecule and inhibit the processes. So this is a direct inhibitory mechanism. First one, direct inhibitory mechanism, and second one is, so this is competitive. Why competitive? Inhibitor is going to compete with the substrate molecule to inhibit the process of pharmacological action or action of enzyme molecule. Now the next one, non-competitive inhibition. So what happened? This inhibitor molecule will attach to the some another location of the same enzyme molecule, which will alter the structure of the enzyme molecule so that substrate molecule will unable to bind to the enzyme molecule. So that inhibitor molecule will alter the structure of the enzyme molecule, as substrate molecule is insufficient. He is not going to bind very easily with that enzyme molecule, and this is a non-competitive inhibition. Now indirect inhibition. Indirect inhibition involves repression and altered physiology. Now what are the types or the repression process of indirect inhibition? In this, decreasing in the enzyme content. See, they are going to decrease the availability of the enzyme, the drug molecules. So if enzyme is not there, there will not be any further metabolism of the drug molecule. And altered physiology due to nutritional deficiency. See, there are patients or the areas where they're not, they will not going to get the nutritional diet, so they may affect, or they have uh, they are unable to produce all the enzymes which are required for the metabolism of the drug molecule. So they have their nutritional deficiency of those enzymes. That's why there will be a hormonal imbalance and they may affect the enzyme inhibition. So such a type of example of the enzyme inhibition process is terfenadine. Terfenadine is the drug molecule. Now, terfenadine is the drug molecule, will be metabolized by cytochrome 3A4, but and convert the terfenadine into the active antihistaminic molecule. But what happens? This erythromycin and ketoconazole drug molecules will inhibit this enzyme, and the conversion of terfenadine molecule into an active antihistaminic drug, the process is completely inhibited. Now, what are the consequences of this inhibition of the enzymatic processes? Increasing in the plasma concentration of parent drug. See, enzymes are very specific in nature and they are going to be stereoselective and stereospecific. So they are going to convert, or they are going to do their reaction conversion very specifically. So what happens if this is a location for the metabolism and where there will be an availability of the terfenadine molecule is a tremendous amount, and that's why this enzyme unable to do his activity of conversion of terfenadine into an active antihistaminic. So there will be an increasing in the plasma concentration of the parent drug and reduction in the metabolite concentration. एकतर जास्त प्रमाणात drug molecule available असेल, then also it is unable to do his activity. Then if the drug molecule is available in too much less quantity, then excreted and prolonged pharmacological effects, then increased likelihood of the drug induced toxicity. Then enzyme inhibition is more important clinically than the enzyme induction. Enzyme inhibition, see, inhibition of the enzyme is more important than the enzyme induction step, especially for the drugs with a narrow therapeutic index. So we have seen both processes, enzyme induction as well as enzyme inhibition. But by comparison point of view, enzyme inhibition is more important clinically, therapeutically than the enzyme induction state. Why? For the narrow therapeutic index drug, because the drug molecules which are going to have the narrow therapeutic index will do or will provide its pharmacological action easily. And the examples of those drug molecules are anticoagulants, antiepileptics, and hypoglycemias. So they will show their action for prolonged period of time because they are going to have the narrow therapeutic index. Now next factors are biological factors. Biological factors are divided into various categories. First factor is age. Now we are going to divide the population into different classes, that is neonates below 1 year, children from 1 to 12 years, and above 12 years we are going to take them into an elderly case. Now what happens in the case of neonates? They have microsomal enzyme system in that neonates are not fully developed. See, we have seen enzyme induction and enzyme inhibition processes, but now we all know microsomes or enzymatic system is required for the enzyme inhibition process or enzyme induction, or microsomal enzymatic system is required for the metabolism of the drug. But in the case of neonates, this microsomal enzyme system is not fully developed. So what happens? Many drugs are metabolized slowly and the half-life. As the microsomal enzyme system is not developed in the neonate, they will require to excrete out the caffeine drug molecule 4 days. But in the case of us or adult people about 12 years, we will metabolize the caffeine molecule within 4 hours. See, this is the difference because the neonates have the neonates didn't develop, the microsomal enzyme system is not developed in those babies. Now the age group within children, that is 1 to 12 years, what happened, the rate of metabolism is reaches maximum because they are in the growing stage and the microsomal enzyme system is too much active. So they will require large milligram per kilogram of the dose than the adult. So the dose of their drug molecule, in the case of children, it is quite high because the now the example of such type of drug is half-life of theophylline in the children is 2/3 of the adult, and the age group elderly, that is in this case there are several factors. They are going to have the reduced liver size, reduced hepatic blood flow, and reduced enzymatic activity. So they are the factors which will affect the drug metabolism. And finally, these are the factors which will decrease the metabolism of the drug molecule. So we have seen already all those factors. Age. Second factor is diet. Now, what type of diet will affect the drug molecule or the enzyme content or enzyme induction or enzyme inhibition process? Now, enzyme content and activity is altered by the dietary components. Now, what are those factors? Now, what type of effect is given by the protein diet, effect given by the fat diet, fruits, and the vitamins and mineral content. So, the effect given by the protein diet, enzyme synthesis is promoted. If we are going to take the protein diet, so the enzyme synthesis in that persons are promoted because we all know enzymes are nothing but they are prepared or synthesized from the protein molecules. Now, increased drug metabolizing affinity. If we are going to increase the production of enzyme, ultimately increase drug metabolizing activity. Now, the second factor is fat-free diet. If we are going to take the fat-free diet, so it will deprocess the cytochrome P450 levels and decreases drug metabolizing ability. Then if we are going to take fruits in our diet, then inhibit the metabolism of the some drug, that is terphenadine. For example, which depending upon the type of fruit, that is if we are going to take grape as a fruit, for example, then this grape will inhibit the metabolism of some drug, and the example of such drug is terphenadine. Now the next is, if we are going to take the vitamins and mineral containing diet, will include vitamin A, B, C, E, calcium, ferrous, magnesium, and zinc. They are going to retard the metabolic activity of the enzyme. So these vitamins and minerals will retard the metabolic activity of the enzymes. Next factor is sex difference. Now, since variations between the male and female, they are observed following puberty. So sex-related differences in the rate of metabolism may be due to the sex hormones. See, in the case of male, the male species, the sex hormones are different, and in the case of female species, the sex hormones are different. So the activity of the drug molecule or the drug metabolism is affected by the factors, that is, this is due to the sex hormones. Such sex differences are widely studied in the rat, where male rats have greater drug metabolizing capacity in human, and women metabolize benzodiazepine slowly than the men. So several studies have been shown that women on contraceptive pill metabolize a number of drugs that is slow rate. For example, studies in the animal. Now, nowadays we are going to use the animals for the study. So when we are going to conduct a study on the male rat, they are going to have the greater drug metabolizing capacity. Now, studies in the humans. In the case of human being, the woman metabolize drug molecule, that is benzodiazepine, very slowly than the men. And the women which are going to take contraceptive pills, they are going to metabolize some drugs slowly. Now, species difference. Species difference is nothing but what type of species we are talking about, whether it is animal or whether it is a human being. So these are the differences. They are mainly due to quantitative, but there are some qualitative differences also. Now, what happened in the case of phase 1 reactions? A metabolism of amphetamine and ephedrine drug molecule. But now we have already seen the metabolism of amphetamine. But in the case of animal species, the metabolism of amphetamine drug molecule will be done by the aromatic oxidation process. And in the case of human beings like us, the metabolism of amphetamine and ephedrine drug molecule will be done by the oxidative deamination process. So this is depending upon which type of species or study is done on which type of species. Now, in the case of phase 2 reactions, variations are mainly qualitative. Now, in the case of glucuronide conjugation, yesterday only we have seen the glucuronide conjugation. So glucuronide conjugation is an important route of metabolism in the mammals, birds, reptiles, and amphibians, but not in the fish, because now we all know glucuronide is easily available, assisting for the metabolism of several drug molecules, because we are totally depending upon the carbohydrate diet, and these carbohydrates are going to produce the glucuronic acid. So easy availability of glucuronic acid will do the phase 2 metabolism study of several drug molecules. But in the fish, this is not the case. But in the case of mammals, cat lack the ability to conjugate phenol with the glucuronic acid. But this process is dominant in the case of pig. So species to species, which type of animal we are talking about, they are going to affect the, or they will change the drug metabolism. Now the next factor is strain difference. Strain difference is divided into pharmacogenetic differences and ethnic variations. Now, study of intersubject variability and the drug response. So in that case, the pharmacogenetics means metabolism of phenylbutazone and antipyrine. Now, this is depending upon the genetic constitution of the organism in which the drug metabolism is done. Now, if we are, for example, if we are talking about the metabolism of a phenylbutazone and antipyrine with the monozygotic twins, from one zygote, if the produced twins are from the one zygote, and if they are twins, so they will very little or no difference in the metabolism of the phenylbutazone and antipyrine in those twins. But if two twins are in the dizygotic, or the twins are from the zygotic twins, so there is large difference between the metabolism of drug molecules. That is genetic variation. They are from the same parental species, that is mother and father. So if the twins are from the monozygotic, there is slight difference in the metabolism. But if the dizygotic twins are there, there is large difference between the metabolism of the phenylbutazone and antipyrine drug molecule. Now the next difference, or the next factor in this strain is ethnic variation. Ethnic variation amongst the variation from the different races. So example of the such drug molecule is acetylation of isoniazide. So what happened? White and blacks will have the equal percent of slow and rapid acetylation found. But in the case of Japanese and Eskimos, for the acetylation of isoniazide drug molecule, slow acetylator dominates. Types of acetylators, rapid acetylators are involved in the acetylation of isoniazide, but in the case of Japanese and Eskimos, slow acetylators will involve in the acetylation of isoniazide. Now the next factor will be altered physiological factor. So pregnancy stage of the woman is the main important factor which will affect the hepatic drug metabolism. Now the physicochemical changes during the pregnancy are probably responsible for the alteration in the drug metabolism. And for example, in the woman, the metabolism of promazine and pethidine is reduced during the pregnancy. Due to pregnancy situation or the changes in the body, they are going to reduce the metabolism of promazine and pethidine. Now, next state or the next factor is altered physiological factors. Already we have discussed about the pregnancy high level of steroid hormone that a woman is going to take when she is on the pregnancy. Now that due to this, the maternal drug metabolizing ability is reduced. As we are going to take the steroid hormones, they will alter the drug metabolism is reduced. Now in the case of disease state, now depending upon that, which or from which disease they are suffering from, like liver disease, renal disease, or cardiovascular failure or myocardial infarction, whether heart problem is there, whether kidney problem is there, or whether the liver problem is there. Depending upon that, they are going to alter the drug metabolism. If liver problem is there, that is hepatitis or jaundice, these are the main associated problems with the liver. So there is reduction in the hepatic drug metabolizing ability. Now the next problem is renal disease, that is oxidation of vitamin D, conjugation of salicylates, they are going to impact. And if the heart problem, that is congestive cardiac failure or myocardial infection, there is decreasing in the blood flow to the liver. So this is all about which I am going to convey you related to the factors affecting drug metabolism. Those are chemical factors, biological factors, and physicochemical properties. Physicochemical properties we have already discussed. These are the main factors which will affect the drug metabolism.